Okay, so in this lecture we talked about just some introductory topics in pathophysiology. We talked about cell adaptation, we talked about cancer, and we talked about burns. So pathophysiology, this is the study of changes in normal physiology that you see in response to disease or injury. This is a bit different than pathology, which is the study of the cellular or the tissue changes that occur in response to disease or injury. A disease is any functional impairment of either cells, tissues, organs, organ systems. It can be any of those, one of those, all of those, and that's what a disease is. A syndrome, this is a specific condition with a recognizable and predictable pattern. The example I gave in class was something like Down syndrome, which is a specific condition and has a recognizable and predictable pattern. Pathogenesis, this is the origination or development of a disease, and then etiology is the specific cause of a disease. Now, etiology can be definite, meaning the exact cause is known, which is often the case for certain infectious diseases and also certain genetic diseases. But it might also be multifactorial or idiopathic, meaning there are many factors that come together or the cause is just unknown. And that's going to be the majority of diseases that you'll deal with. Clinical manifestations, these are the objective signs that you see with a disease. So these are the things that you can see or measure. Symptoms, of course, these are subjective, meaning they're reported by the patient. So things like pain and headaches and nausea and all of those types of things, those are symptoms. Clinical manifestations, they can be acute, meaning rapid onset. They can be chronic, meaning more long-lasting. The book gives the value of greater than 6 months. Or they can be subacute, which kind of falls in between acute and chronic, oftentimes defined as 3 to 6 months. They can be insidious, meaning the onset is you can't really pinpoint when things started. It kind of came on slowly. And of course, they can be local or systemic. Morbidity, this refers basically to having a disease or a negative outcome with disease complications. And this can result in mortality or death. So when we talk about prevention of disease, it can be primary, secondary, or tertiary. Primary prevention prohibits a disease from occurring in the first place, like wearing a helmet. Secondary prevention, this is basically early detection of a disease through screening, so things like colonoscopies or pap smears. Tertiary prevention, this is rehabilitation after a disease has occurred. So examples are different types of physical therapy and other types of therapy. So a quick review on cells. Cells are the basic structural and functional unit of the body. 3 main parts are the nucleus, plasma membrane, and the cytoplasm. Now cells are able to adapt, and they adapt in order to try to maintain homeostasis. And adaptation is important, but it's not effective against long-term stressors. Okay, and if long-term stress is present, cells can become injured, which may be reversible, but if it's irreversible, the cell will die. And there's a bunch of ways that cells can become injured. So let's talk about cell signaling. So basically, cells have to talk to each other. And there are 3 main mechanisms by which they do that. Paracrine signaling is when cells will produce molecules that affect nearby cells. Things like growth factors, clotting factors, those are important paracrine signalers. And also allergies, other components of the immune system operate this way. Autocrine signaling, this is when a cell produces a signaling molecule that will affect itself. And this is pretty rare, but you see it a little bit in the immune system, especially with like T cells and things like that. Endocrine signaling, this is when these signaling molecules are released into the blood, and these are called hormones. And these are going to affect tissues in distant sites. And we'll talk about all of the endocrine system in a couple of lectures. So like I said, cells can adapt, and that's important because they need to maintain homeostasis. So cellular adaptation, reversible structural and or functional response both to normal and abnormal physiological conditions. So of course this can be normal or associated with pathology. Now the first one we're going to talk about is atrophy. This is when you have a decrease in cellular size. And this can be physiologic, can be normal in the case of involution of the thymus. So the thymus will atrophy as we age because we just don't need it. Or it can be pathologic, like in the case of disuse atrophy of skeletal muscle. Hypertrophy, this is the opposite. So you have an increase in cell size due to signals, growth signals, and these are called trophic signals. And there's a number of these in the body. Things like different hormones, like testosterone, insulin, these are trophic signals. Can be due to increased workload, so like exercising your muscles causes hypertrophy. The muscle cells get bigger. Hormonal input, you see this with reproductive cells, both sperm and egg, or an increased functional demand. So, for example, the example I give here is adenoid hypertrophy, and this is when there's an immune response in the adenoids, which are some of the tonsils, and that increased functional demand causes them to increase in size. But also you see this with, if the heart is overworked, people with hypertension often have hypertrophy in the ventricular wall, so just an increase in cell size. Hyperplasia, this is an increase in cell number. And again, this can be due to an increased workload, due to hormonal stimulation, which you see in the endometrium in the monthly cycle, or in pathology such as benign prostatic hyperplasia. So this is just showing some of these examples here. So here are some normal cells. Here are cells that have undergone atrophy. They've gotten smaller. Here, cells have gotten bigger. That's hypertrophy. And then here you can see that they have increased in number, which is hyperplasia. So metaplasia. Metaplasia is when one type of cell changes to another type. Okay? And a couple of important examples here. One is called bronchopulmonary metaplasia. This is when the pseudostratified ciliated columnar epithelium of the respiratory tract changes to stratified squamous. So we change from one type to another. And also Barrett's esophagus, which is seen in people with chronic GERD. And that's kind of the opposite. You have squamous cells that change to a type of columnar. Metaplasia can be associated with cancer, but it doesn't indicate cancer. Okay, so if you see metaplasia on a microscope slide, it doesn't mean cancer is present or that it will develop, but it's not something you really want to happen. And then dysplasia, you see this with especially long-term or severe stressors on cells. And here you have just changes in the size of cells, their growth pattern, the shape of the cells, and again, it can precede cancer, like in cervical dysplasia, but it doesn't indicate cancer. So here we can see metaplasia and dysplasia, normal epithelium. Here we can see that it's changed from one type to another, that's metaplasia. And this is dysplasia. Dysplasia, you have this very random kind of growth pattern. You have cells in different stages of the cell cycle. You have big cells, small cells, cells with that are multinucleated. So dysplasia is not really a place anybody wants to be. And this is just the example with Barrett's esophagus. And these are all of our adaptations all on one slide. Okay, so make sure you know the characteristics of each of those types of adaptation. And this is actually a really good concept map. It's really simple, but it kind of sums everything up. So a normal cell under stress will adapt. If it does not adapt or does not adapt well enough, you have cell injury. If that cell injury is mild or is temporary, meaning the stressor is removed at some point, it's reversible and you can go back to a normal cell. If that cell injury is not mild or it's not temporary, you'll have irreversible injury and the cell will die. And these are examples of diseases that involve cellular adaptation. There's many, many more. I mean, most diseases are going to have some form of adaptation, but we're going to talk about these later in the course. So let's talk about cell death. Again, this is when cell injury is irreversible. And we're going to talk about apoptosis and necrosis. Apoptosis is typically normal. It can be associated with pathology like infections, but typically normal. And this is programmed cell death, meaning it's genetically controlled. And this is going to occur typically on a smaller scale than necrosis. Okay? So this is programmed cell death, sometimes called cell suicide. Now necrosis, this is when you have the death of cells due to more extensive cell injury. Okay, so the cells are injured. In apoptosis, the cells aren't necessarily injured. They might be a normal cell, but they're programmed to die. Now, necrosis typically happens on a larger scale than apoptosis, and it's not normal, and it often occurs due to a lack of blood flow. So this is just showing some of the changes associated with necrosis and apoptosis. Now, some mechanisms by which cells become injured. You have mechanical injury. This is direct impact or direct trauma. Thermal injury. This is due to extremes of temperature. So examples of that are like burns and frostbite. Toxic injury. Toxin chemicals, they can be endogenous, meaning from our own body, or exogenous. And deficit injury. This is when cells are deprived of something they need, like oxygen or glucose or water. So let's talk a little more about this hypoxic injury. And the most common cause of hypoxic injury is ischemia. Remember, ischemia is the lack of oxygen due to a lack of blood flow. And this can be due to the narrowing of arteries, the blockage of blood vessels, oftentimes both. So I talked about this a little bit in class. When we don't have oxygen, we need oxygen in order to make energy. So if we're not making energy, we can't power things like the sodium-potassium pump. So when a cell is deprived of oxygen, the sodium-potassium pump can't be powered because it doesn't have energy. So sodium moves into the cell, water moves into the cell, so the cell is going to swell up, potassium leaks out of the cell, and a bunch of other changes occur inside the cell. Now, oxidative stress. This is when reactive oxygen species like free radicals, oxygen radicals, will overwhelm our antioxidant systems. So a free radical is an atom or a molecule that has an unpaired valence electron, and that makes it unstable. And these free radicals can cause a lot of harm to almost every macromolecule in the cell, such as proteins, nucleic acids, and lipids. Nucleic acids, these are things like DNA. So you can see how this can be a problem because everything's controlled by the DNA. Now, free radicals are normal. Reactive oxygen species are normal, but we have ways to combat them. And these are called antioxidants or antioxidant systems. So here's just kind of showing some of these free radicals like superoxide and hydroxyl radicals. I don't really care so much that you know this. Just know that if we have too many of these reactive oxygen species, they can overwhelm our antioxidant systems and can cause lots of damage inside our cells, and it can lead to things like cancer in some cases. So infectious injury, this is due to the pathogenicity of a pathogen. It can be caused by direct destruction of cells by pathogens, the production of toxins by pathogens like bacteria, or even hypersensitivity reactions. Now, a lot of times pathogens don't harm us directly, but our immune system, our own immune system, is the one that's doing the damage, and that is immunopathology. Okay, then we spent some time talking about cancer. Cancer is just abnormal growth or division of cells that have the ability to spread. They have the ability to metastasize. Now, a tumor is the same thing as a neoplasm. Neoplasm just means new growth, and tumors can be benign or malignant. The difference is malignant tumors have the ability to spread. Now, benign tumors can become cancerous in the case of things like polyps, but it doesn't mean they're going to become cancerous. So some nomenclature here. Cancers are named based on the tissues that they come from. So, for example, carcinomas arise from epithelium, so like squamous cell carcinoma. Sarcomas come from connective tissue, like osteosarcoma, which is a type of bone cancer. Lymphomas arise from lymphocytes, and they typically will be located in the lymphatic system, like the lymph nodes. And then leukemias arise from the blood-forming cells in the bone marrow. So how does cancer get started? The big takeaway here is that it all starts with the DNA, and it takes multiple DNA mutations in order to get cancer started. Okay, that's the key to all of this. And these mutations, for some reason, give cancer cells a competitive advantage over normal cells in the vicinity. And once these normal cells become transformed, once this cancer ball starts to roll, they have special characteristics. One is they don't have such a need for growth factors, meaning they can grow in the absence of growth factors. Most cells need to be told to grow. Cancer cells don't need that. They also have immortality, which means they don't die. which is, really dangerous. They also have a lack of contact inhibition. So remember that if this is our basal lamina, and we have cells here, when these cells grow, for example, when these cells grow this way and these cells grow this way, and they touch each other, they stop. That's normal. Cancer cells don't listen to these signals, so they just keep growing on top of each other. And you can see how that would be a problem. And they also have no anchorage dependence, meaning they don't need to be anchored to something like the basal lamina or to other cells. So that gives them the ability to spread. So this is just kind of showing that this all gets started with multiple mutations in the DNA. Okay, it takes multiple hits. There are rare cases where it only takes one, but those are genetic, truly genetic types of cancers. For most cancers, it takes multiple subsequent mutations in order to transform that cell to a malignant cell. And these are some of the ways in which cancer cells are able to kind of continue to grow, and their growth goes unchecked. And these are all areas of research at the moment. And we're going to talk about some of these. The ones that we talk about specifically next, those are the ones you should be familiar with. You don't need to know this whole circle. So the genetics of cancer. Like I said, cancer at its core is based in the DNA mutations, multiple DNA mutations. And some of these genes that are involved with cancer have specific categories. For example, proto-oncogenes. These are genes that normally control the growth and the division of cells. Now, say one of these proto-oncogenes develops a mutation for some reason. Once they develop a mutation, we call these oncogenes. And these are genes that can now cause malignancy if a mutation occurs. Like I said, cancer takes multiple mutations. So if a proto-oncogene gets one mutation, it doesn't really mean much. But once it gets that mutation, now you can call it an oncogene because subsequent mutations increase the likelihood of malignancy. Tumor suppressor genes, these are genes that inhibit the growth and the cell cycle of cells. I talked about the p53 gene. You don't really need to worry about that. But if you have mutations in tumor suppressor genes, you can lose the regulation of cell growth and cell division. This is just showing some of these proto-oncogenes and how things can kind of happen. You don't need to worry about that. So genomic instability. This is basically the increased tendency for mutations during the cell life cycle. Our cells are not immortal. And, many of our cells live a long time. And as we age, we develop more mutations. Cancer can be seen as a disease of aging. But we do have protective measures to try to make our cells last as long as possible, but they're still not immortal. Telomeres, these are like little hats at the ends of the chromosomes. And what they do is they protect the chromosome. And they are made by an enzyme called telomerase. However, as we get older and cells divide, these caps get shorter, and shorter. So our chromosomes get less and less and less protected. Cancer cells can actually activate this enzyme telomerase. So they can keep making new caps, and that allows them to continue to divide and be immortal. So this is just a little micrograph showing these telomeres, which are these little caps. So angiogenesis. Angiogenesis is the formation of new blood vessels. And tumors need a rich blood supply. They need a rich glucose supply. And cancers, they are able to secrete factors that stimulate angiogenesis. Some of these are VEGF, which is vascular endothelial growth factor, or PDGF, platelet-derived growth factor. So that's another way that cancer can kind of continue to grow, is because they can stimulate the formation of their own blood supply. So chronic inflammation. Persistent inflammation is an important factor in many diseases, including cancer. And, diseases like diabetes and obesity and certain lifestyle factors like smoking and drinking, these are all pro-inflammatory, and they're all risk factors for developing cancer. Also, when you have inflammation occurring in the body, you're going to have immune cells, which we talked about last quarter, And you have these macrophages, which are called TA macrophages, or tumor-associated macrophages. And they are found in the framework of tumor cells. And they can consequently promote tumor survival by blocking the activity of certain T cells, like natural killer cells and cytotoxic T cells, CD8 positive T cells. And nobody's really it's not understood why macrophages do this, but they do. So that's a reason why inflammation can be really harmful. It's just one factor. This is just showing these tumor-associated macrophages and the signaling that they do with these other immune cells. You don't need to worry about this, though. So, of course, the immune system keeps us safe from pathogens, but it also can undergo immune surveillance. It can seek and destroy cancer cells. So, of course, immunosuppression in any way, whether through disease or disease treatment, increases the risk for cancer. And viruses are also associated with cancers, like the hepatitis, viruses B and C, the chronic types. They're related to hepatocellular carcinoma. EBV is related to a couple of cancers like nasopharyngeal carcinoma and Burkitt's lymphoma, and also HPV with cervical cancer. So this is just another little picture showing how the immune system kind of works when it comes to tumors and cancer. So this is showing, this is clearly a significant type of cancer here. And HPV strain 16 and 18 are the most commonly implicated strains when it comes to cervical cancer. You don't need to worry about that, though. So metastasis. Remember, this is a hallmark of cancer. It has the ability to spread. And metastasis is the spread of cancer from an original site to another site. Local spread, this is just when nearby tissues or organs are invaded. But when these cancers spread via the lymphatics or the blood to distant sites, that's associated with advanced disease, meaning this cancer is, it's stage 4. Now, as cancer cells begin to dedifferentiate, meaning this is where I talked about the definition of anaplasia. Well, I talked about anaplasia when we talked about grading cancers. When they dedifferentiate, meaning they become less and less and less mature, they become more stem cell-like. They are more able to metastasize. And certain cancers will metastasize to different sites. So clinical manifestations of cancer, it's going to be based on the location of the tumor, the type of the tumor, the size of the tumor, and many early manifestations of cancer are pretty vague. A lot of people are asymptomatic, unfortunately, until the cancer is more advanced. So pain can be due to destruction of tissue, can be due to pressure placed on different tissues and organs by the tumor, stretching of tissues or organs, obstruction, sometimes infection. Fatigue, by far the most common symptom. It can be due to treatment, it can be due to the psychosocial sort of state of the patient, due to the fact that they have cancer, poor nutrition, due to, not having an appetite because of treatment. So fatigue it's due to more than just the disease itself. So cachexia, this is wasting, specifically wasting of muscle and fat that you can't replace. So it can't be nutritionally corrected. So you see this with cancer, you see this with AIDS. Anemia, this can be due to bleeding, of course, but also nutritional deficiency, as well as cancers of the blood cells themselves, and things like chemotherapy is pretty toxic to bone marrow, so a lot of times you will see anemia, as well as leukopenia. So leukopenia, which is a low level of white blood cells, again, this can be due to chemotherapy, or even a tumor that has invaded the red bone marrow. And of course, infection, because these people are immunocompromised. So cancer diagnosis, imaging, so CT scans, PET scans, MRIs, X-rays to kind of see, you'll at least see a tumor. Not necessarily, but it's a way to visualize tumors. A biopsy, this will confirm cancer. There are also tumor markers, different hormones and enzymes, and even genes that you can look for. These are not diagnostic. They aid in diagnosis, but biopsy is really the way to confirm whether or not a particular lesion is cancerous. So the staging of cancer, this is based on the extent of the tissues involved for a particular tumor. So in stage 1, there's no metastasis. It's very localized. So this is stage 1 here. Okay. And you can see that this tumor, this is colon cancer. This tumor has invaded a couple of the layers of the bowel wall. It has one lymphatic that's kind of draining this tumor, but still pretty localized. And if it's caught at this point, the chance of survival is pretty good. Now, stage 2, we start to have more local invasion. So we're starting to get deeper into the bowel wall. We have more of these lymphatics draining the tumor. We have more blood vessels that are supplying the tumor. So this one's a little more significant. In stage 3, we start to see regional involvement. So in this case, we have regional lymph nodes that are involved. So this lymph node has cancer cells in it. And then finally, stage 4, this is where we have metastasis to a distant site. So it's spread to another part of the body. I mentioned the TNM system, which is another way to kind of classify cancer. Don't worry about it. I just wanted to point it out in case you ever come across it. So treatment of cancer, of course, surgery, radiation, chemotherapy, but there are some new kids on the block that are going to be really the future of cancer treatment. So surgery to either remove precancerous lesions like polyps, to obtain biopsies, to remove lymph nodes, to remove the tumor itself, of course, and sometimes for palliative care. Radiation therapy, this is basically using ionizing radiation to directly damage the DNA of a tumor. And radiation therapy has gotten a lot better. It's a lot more focal, so we can shoot a more direct beam of radiation, but there, of course, are risks. And then chemotherapy. The goal of chemotherapy is to kill cells that are dividing very rapidly. Cancer cells divide very rapidly, so they're a decent target. However, there are many other cells in the body that divide rapidly. For example, the cells of the hair matrix, which make new hair. Those are targeted, so that's why people's hair falls out. Cells of the GI tract, like the stomach, divide very rapidly, so those cells are targeted. Cells in the bone marrow are targeted, so it's not a really specific. It's gotten better, but it's still not ideal at all. And then immunotherapy. This is going to be the future. The future is going to be things like personalized cancer care, where people's immune cells can be trained to target cancers, and that's been shown to have promising results, but it's very expensive and it's not widely available. And of course, vaccines to even prevent cancer, like cervical cancer. So that's kind of the future. Okay, then we talked about burns. So there are first, second, and third degree burns, and I threw in fourth degree burns. So a first degree burn, this is a superficial partial thickness burn. Anytime you see partial thickness, that just means it doesn't go all the way through the skin. So first degree burns only involve the epidermis. These are like sunburns. So you'll see the redness, the erythema, mild to moderate pain. So your typical sunburn. Second degree burn, this is a deep partial thickness burn. So this goes all the way through the epidermis and into the dermis. And here we see the formation of blisters. And in blisters, fluid accumulates, this exudative fluid, and it will basically separate the dermis and the epidermis. Now the follicles, the hair follicles, and the sebaceous and sudoriferous glands are going to be relatively undisturbed, depending on the severity, because of their depth in the dermis. So they're relatively undisturbed. Now a third degree burn, this is a full thickness burn. And in a full thickness burn, it goes all the way through the epidermis, all the way through the dermis. That's why it's full thickness. And oftentimes will go into the subcutaneous layer, the fat. Now afterwards, it's generally painless because all of the nerve endings have been destroyed. And in this case, you might need a skin graft in order for it to be repaired. And then fourth degree, again, the book doesn't talk about this, but this is even deeper. This is when you start to destroy fascia and muscles and even the bone. So these are really significant burns. Then I talked about the rule of nines. I just mentioned kind of how it can be used, but you really don't need to worry about it. It's just something I wanted to point out. So as always, if you have any questions, please let me know.